Okay, great. So yeah, hello there. Sorry I can't be there, but one of my kids has their birthday today, so that was a very good reason not to be able to travel. Um, so I will tell you a bit about what we do in the lab, but not about any of the Drosophila fly part of it, but all organoids and what we find interesting there at the moment. But as an introduction, kind of overall, what we're interested in my lab is we want to understand how organ shape and therefore function is encoded by the genome. And so at the beginning of their development, really all embryos and also organ systems start off from very simple structures that are then very quickly diversified transcriptionally. And this transcriptional patterning is then turned into physical changes at the molecular, cell, and tissue scale. But in many cases, we do not understand the connection between the two very well. And so that's really what my lab wants to unravel, what is the initial transcriptional blueprint that needs to be put in place to kickstart the physical changes, what are the mechanics of these physical changes, and then is transcriptional control really the key to the whole process, or are there other mechanisms? So one of the systems we use to study this is the formation of the tube of the nephron, of the kidney nephron in human renal organoids in culture. And so here really the first step of the tube morphogenesis, or the formation of the tube, is really preceded in this kidney organoids by a process of cell polarization. Before then, a narrow lumen tube starts to form and expands, eventually differentiating into the different sections of the nephron. And so to understand morphogenetic processes like this, what we do in my lab is, or we know we need to address mechanisms really at multiple scales of magnitude. And so this is what my lab is doing and has done over many years in different systems. So what we do is we look at questions at all scales of magnitude, such as transcriptional patterning that is occurring, the function of molecular components, how cellular changes feed into the process, and how such cellular changes are then coordinated at the tissue scale. And so I want to tell you now about some of our recent findings addressing the formation of the early nephron tube and also explain the system, the culture system of the organoids a bit. So the work I'm showing you was done by an amazing postdoc in the lab, JP, and the second part I'm talking about by a very talented PhD student, Rob, and it's in part a collaboration with AstraZeneca and Julie Williams in their renal department, and also a collaborator here in the University of Cambridge. So nephrons really form the functional unit of the adult kidney, and they form in vivo through an interaction of the so-called ureteric bud that forms from an initial epithelium, or buds off from an initial epithelium called the nephric duct. And the ureteric bud invades a tissue called the metanephric mesenchyme, so mesenchymal unpolarized cells. And through interactions, oops, I'm very sorry, I'm moving my mouse too much. Through interactions between the two, this leads to aggregation of cells and then initial polarization, a mesenchymal to epithelial transition, before then this yellow tube here formed initially from the mesenchymal cells expands and fuses with the ureteric bud-derived structures. And everything from the ureteric bud will really just form the collecting duct system later, but the nephron is formed from these mesenchymal cells. So each of us is born with between 200,000 and over 1 million nephrons in our kidneys. They're all formed prenatally, and the nephrogenesis really is recapitulated very well in this renal organoid system we're using, which I hope I will convince you of. So really, as I already briefly said, the first highly coordinated step in nephron morphogenesis is this process of cell polarization, or mesenchymal to epithelial transition. This forms this initial so-called renal vesicle of cells, a polarized epithelial structure, then immediately begins to expand into a narrow lumen tube. And so in the lab, we can derive human renal organoids from induced human pluripotent stem cells. And then throughout the protocol between day 10 and 14 in culture, these induced cells then undergo this mesenchymal to epithelial transition before forming the nephron. So there are three aspects I really want to highlight about this protocol that I want to point out. But actually, first, I should say the way this protocol works is that during the initial steps, treatment with a Wnt activator, chemical called chiron, which is a GSK3 inhibitor and therefore drives Wnt signaling activation, really drives the cells towards what is called intermediate mesoderm fate, which is what cells go through in vivo on the way to developing into kidney. And then more Wnt activation together with FGF9 pushes these cells really towards nephron fate. And so the three things that are important about the organoid protocol we're using, which is a variation of a protocol initially developed by Melissa Little's lab. So there are three important things. In our organoid protocol, what we do not form is anything that resembles the ureteric bud-derived structures. And so these usually provide the Wnt signal important for the aggregation and polarization of cells. And so therefore we provide that in the culture medium, and it has the same effect on these mesenchymal cells that are our starting point. The second important aspect is that we start off from iPSC, so stem cells in a 2D culture in a dish, but then between day 7 and 10, we move the cells into these aggravell plates. So we use 1,000 cells, and they will then over a day already aggregate into a ball of cells. So we're going from a 2D culture system into a proper 3D system. And that is really important. And then the last point is that the spheroids are then cultured in a suspension shaking culture. And from day 12 on, there's really no further growth factors or signaling factors or anything that directs their morphogenesis. From that point on, everything happens intrinsically within the organoid. So once you've pushed them far enough into the right direction, they know what to do. And so it's all self-organization from this point. And just to point out again what the types of cells are we can generate in this version of the protocol, and there are different versions available that other labs use, but we are interested in the formation of the epithelium and the early tube. So this is a schematic taken from a recent review by Melissa Little, actually, which shows all the cell types that are eventually generated in a kidney in vivo. And so in our protocol, what we really do, we drive cells down this path highlighted here by the red arrows, which is really metanephric mesenchyme, and then induce cells that form the renal vesicle and the different cell types that are in a nephron. But we don't form, as is shown here, ureteric bud and the whole collecting duct tree. We sometimes get differentiation of some endothelial cells that also can arise from these early precursors. But they're very cell types that we do not form in this protocol. So what the cells do in culture, I already said, this mesenchymal to epithelial transition initially before the morphogenesis of the tube really happens is characterized really by this acquisition of epithelial polarity, which you can see here with classical polarity markers, APKC, atypical protein kinase C for the apical side, and integrin beta 1 for the basal side. And so once this polarization, or MET for short, is underway, a narrow lumen tube begins to expand, which is really the beginning of the nephron. So similar to what we've done, well, first of all, just in terms of polarity, so there are many different complexes and aspects of epithelial polarity that have been defined in many systems. So we have many good markers we can look at to follow the process and progress of the epithelialization. And really different aspects of these complexes that interact with each other can be important for cell polarization. And in fact, I've just been at another conference last week that was all about these two possibly reverse processes of cell polarization, mesenchymal to epithelial transition, or epithelial to mesenchymal transition, so going from a polarized to an unpolarized state. But it is still not completely clear how these are organized in terms of the transcription factors driving it, and in terms of what sort of cell states are in between a fully polarized cell and a mesenchymal cell. So that's something we're really interested in our system. How do you get, which path really do you take to go from unpolarized cell to a polarized epithelial cell? And can we control this? Because this might be important later on in the future in terms of understanding repair processes in the kidney better. So similar to other approaches we've taken, for instance, in the fly, we wanted to understand really what is put in place early on in terms of a transcriptional blueprint that kick-starts the morphogenesis, and in the case of the nephron, really the polarization of cells. And so we across the time course of polarization and early expansion of the tube, we established this initial blueprint through multi-omic sequencing. So we did either just single-cell RNA sequencing or a combination of single-cell and ATAC-seq, which can show you where chromatin has opened up and reveals which genes might be active. And we did this across the time course of the protocol. And so what this showed us initially, first of all, is that overall we can identify all the different cell types of the nephron that we would expect in the type of organoid protocol we're using. And we're very lucky because in my institute, in one part, is a bit of the immunology department of the university here, and a very good collaborator, Menna Clatworthy, who's a clinical nephrologist, mostly interested in immune cells, but together with collaborators, she and other labs recently generated a single-cell RNA sequencing atlas, actually of human fetal kidney samples that they had access to through hospitals. And so we could compare our organoid sequencing data to these real in vivo fetal data to see how similar, at least transcriptionally, really, the organoids at early stages are. And so we could, this showed a real strong congruence between our two data sets. So here what we do is we superimpose our organoid sequencing data on top of the UMAP of the fetal data, and can see that over time, really, our data move along the same sort of timeline as they could see in the fetal renal system. So this was really reassuring. So if we then cluster our multi-omic sequencing data and generate that covers this period of the mesenchymal-to-epithelial transition, which is between day 10 and 14 in culture, then we see that splits into really clear lineages, the stromal lineage and the epithelial lineage, and also the different time points throughout the progression and polarization. And when we then analyze these clusters for marker genes that are expressed, that really define these different clusters, then they really highlight a progression through that really demarcates these cells becoming more and more epithelial. But it also shows, if we look at enriched Go terms in these different clusters, that really what is switched on is cell adhesion modules, things that control the cytoskeleton and control morphogenesis. So things really that help cell-cell coordination, cell shape changes that are important, not surprisingly, in this process. So what this analysis also allows us to do, the paired single cell RNA-seq and ATAC-seq, allows us to look at transcription factors that might control the process. And we can group these into different classes because we can look at transcription factors that are highly expressed and whose motifs, where they bind across the genome, seem to be opened up as shown in the ATAC-seq data. So those would be activators potentially, because they will be able to bind and therefore hopefully be able to activate downstream genes, whereas factors that are expressed but where chromatin kind of is not accessible might have been repressors that are bound potentially, but have not led to opening up of chromatin, so maybe have actually lead to shutting down of accessibility. So if we now just look at epithelial activators that we identified in the data set, then one particularly caught our attention, a transcription factor called PAX8, that together with PAX2 had been in mouse studies implicated in nephromorphogenesis, but actually in mouse it seems PAX2 plays an important role, whereas I will show that we think in the human nephrogenesis timeline, PAX8 really is a key transcription factor. So if we look at, and PAX8 caught our attention because if we look at the protein level, and PAX8 is expressed and present really early in nuclei of cells in the organoids before, so these are all the same organoid stained with 4 different labels, before any markers of nephron fate, such as HFN1b or E-cadherin, so a marker of starting polarization of cells are seen. And it's there really early on before any of the MET or morphogenesis happens. And so what we can then do in the organoid system is use CRISPR-based interference to eliminate PAX8 function in the renal organoids. And this is done through a combination of plasmids we get into the organoids that have a guide RNA targeting the right locus, and then, oops, I'm sorry, no, that's too fast. And then a Cas9 that is fused to a repressor domain called CRAB. So this will then bind your target gene of interest and repress transcription. And this works very well. If we look at the RNA levels, there's a really good reduction of PAX8 at the RNA level using this system. And so what happens then in the organoids? So this is a control organoid where at day 14, a large convoluted tube section has formed that's positive for PAX8 and E-cadherin as one of the markers of an epithelial state. And if we now knock down PAX8, what happens is that in knockdown cells that are marked with GFP here as well, we do not see any tube form, only this small region down here that is GFP negative and seems to be cells that have escaped the knockdown. They still express PAX8 and then they also still switch on E-cadherin. So you need PAX8 to initiate the mesenchymal to epithelial transition, it seems. And so to test or to make sure that this is specific, we use different guide RNAs to make sure we target the right locus. But we also tested if we now re-express PAX8, can we rescue this defect? So here again, we knock down PAX8 and all the green cells are knockdown cells. And as I just showed you, there's a small bit of tube that can form, but it's all formed from GFP negative cells, so they have escaped the knockdown. And here we just re-express an empty lentivirus control. But if we now have a lentivirus to re-express PAX8 in the knockdown, what we can see is that now the forming renal vesicle tubular structures here have many of the GFP knockdown cells that initially lost PAX8, but they're now re-expressing PAX8 and therefore they can reincorporate into tubes. And in fact, all PAX8 overexpressing tubes, either knockdown organoids or wild-type ones, get incorporated into tubes, really suggesting that this transcription factor drives cells that express it at high enough levels down this fate choice to epithelial and nephron fate in these organoids. So, but what does PAX8 do then to drive this? So if we look at basically a footprint of PAX8 across the genome, across the time frame of the polarization, and we can do this using the attack data, seq data, then we see already at day 10, but continuing throughout the whole time period of the polarization, there's a strong peak of PAX8 binding at near transcriptional start sites, and therefore most likely activating gene expression. And we can now use our attack data to identify, we can correlate accessible regions or peaks that we identify in the attack-seq data to co-regulated expression of nearby genes, which identifies possible regulatory regions and use this really to, and then look for predicted PAX8 binding sites in these opened up chromatin regions. So this really allows us to define a PAX8 regulon, or things that are regulated and controlled transcriptionally downstream of PAX8, and identify that PAX8 really seems to drive a cell adhesion program. So cadherin 6, CDH6 has opened up chromatin regions with PAX8 binding sites. The same is true for E-cadherin, as well as, not surprisingly, factors such as HFN1B, which is a transcription factor that's key to later nephron segment differentiation. And if we actually look at the levels of several cadherin genes across the time period of the MET polarization, then we can see that their levels are very tightly controlled across this time course, and they're not expressed in the stromal or mesenchymal cells at all. So we can also use our data and perform a so-called pseudotime analysis of the multi-omic data. So this allows us to kind of derive a more continuous timeline of changes. So for example, I'm showing you here again expression of two cadherin genes, EpCAM and E-cadherin, and you can now see that across just the day 10 to day 14 mesenchymal to epithelial transition, these two cadherins both increase, not surprisingly, in their expression. Well, we can use this to analyze the transcription factors that might control the MET a bit more closely. So if we just look at activators, then there's some activating transcription factors such as TCF7 or LEF1, which are transcription factors that are downstream of the initial Wnt signal that we need to actually activate overall nephron fate in the mesenchymal cells already. So these two are expressed highly at the beginning of the polarization, but then become downregulated, and so is the accessibility of their motif in the genome. Whereas factors such as HNF1B that I already briefly mentioned becomes expressed more and more, and its binding sites across the genome become more and more accessible, because this is really needed later on for nephron segment differentiation. So I just showed you in the slide before that Wnt signaling output, the two transcription factors downstream of Wnt activation become downregulated over the MET. And we knew actually from the literature that Wnt signaling really seems to need to be attenuated for proper nephron development, but it was really unclear why. But our organoid model now allowed us to test this, because what we could do is usually at day 12 of the protocol, all growth factors are withdrawn and there's no Wnt activation or anything else in the medium any longer. But if we now keep the Wnt activation on for longer, we can have a look at what happens to the cells and the tubes. And in fact, if we do this compared to a control where there's a nice polarization with cadherin, and Z01 is an apical marker here nicely, the center of the tubes, when we keep Wnt signaling on for longer and look, then the cells have lost polarization or never reached a very good state of polarization, similar to how they looked at much earlier stages of the protocol. And as a control, if you combine this excessive Wnt activation with a Wnt inhibitor, then this looks like the control situation. So really, you need to switch Wnt off throughout the polarization to reach a proper epithelial state. And we can also see this if we look at expression of various epithelial genes across the normal three time points we analyzed, and then here in light blue is now when we keep Wnt signaling on, you can see all these epithelial genes are much lower expressed than they should be. So in terms of the transcriptional control really of nephromorphogenesis and tube expansion, we think there's an early Wnt signal that is well described that we think possibly directly activates PAX8 expression, which is then key to activating a transcriptional program, switching on cell adhesion and cytoskeletal factors that drive the MET, but then to complete the polarization and maintain an epithelial phenotype and mature junctions, for instance, Wnt needs to be deactivated, because otherwise factors such as HFN1B or also T transcription factors are not expressed. And so these are important for nephron segment differentiation, but also linked to potential mechanotransduction and control of the whole process. And that's something we're actively looking at. And I just want to briefly tell you a bit about looking really at the cell biology of how MET is implemented at the molecular, cellular, and tissue scale. So what really defines this polarization is that the signal coming from the apical or basal domains of cells and how is it coordinated between the cells. So we know from culture cells, 3D culture models, but also many different model organisms that different cases, both apical and basal cues can lead to polarization. And in our case, in the nephron, we can think of cell ECM interactions, for instance, driving the cell polarization, or it could be driven by cell-cell interactions. And I already just showed you that PAX8 really seems to switch on the cell adhesion program by controlling directly potentially cadherin 6 and cadherin 1. And if we look at all the cadherins we can detect early in our sequencing data set, then really CDH6, cadherin 6 seems to be the one that is at day 10 already, at the start of polarization, much more expressed in the induced cells. And we can also see this at the protein level. If we look at antibody staining for many different cadherins, then really cadherin 6 at day 10 is already present and is only present in the induced cells that will form the nephron tube. And E-cadherin, for instance, CDH1, comes on a couple of days later in the protocol. And we can see if we zoom in to the organoids that at day 10, in the induced PAX8 positive cells, CDH6 is expressed but not polarized, but only a few days later it is beautifully localized to these forming apical domains in these renal vesicles on early tubes. And if we zoom in even more, then really early on, CDH6 is in the plasma membrane, but unpolarized, and then becomes restricted to the forming center of a tube in the apical domain. And so again, we can use the CRISPR-mediated knockdown to see if it's required in the organoids. So in the control, we form nice tubes, but if we knock down CDH6, again, we lose most of the tubes. So really, CDH6 seems to be important also downstream of PAX8 for this cell polarization. But what about integrins or extracellular matrix, basal lamina, that also has, in theory, the possibility to polarize cells? And if we look at this here, laminin in the top row or integrins, the bottom row in comparison to, for instance, Z01, which is an apical domain marker, then you can see Z01 seems to concentrate and aggregate into forming apical regions before we ever see any real condensation of either laminin or integrins and forming basal sites. So we think there's a temporal delay. Although then after a few days, laminin and integrins beautifully co-localize in this forming basal lamina that by day 14 then has formed a really nice shell around the forming tube in the organoids. But if we knock down integrin beta 1, for instance, and this is still preliminary, but at the moment we only have fairly mosaic organoids for this, we can see that in individual knockdown cells here, where we can see integrin beta 1 is missing basally, these still at early stages at least integrate into forming tubes. And still then these knockdown cells seem to also have apical markers present. So we think integrin beta 1 at least might not be required. So our model at the moment is that CDH6, which is expressed really early at a stage when induced cells still show salt on pepper sort of pattern in the organoids, helps these cells to aggregate into clusters. And then this aggregation leads to the formation of early forming apical domains that potentially physically help to exclude integrins that are also already present in the plasma membrane but are not polarized. And this overall then coming from the apical side leads to a polarization and the formation of a bipolar polarity vector that really defines this epithelial apical basal polarity axis. So I hope I showed you that renal organoids derived from human-induced pluripotent stem cells provide an excellent human developmental model of mesenchymal-to-epithelial transition. And I hope I convinced you that PAX8 is a crucial transcription activator of this MET program. And then really in human nephron formation and MET, the cell-cell adhesion that is driven by cadherin 6 seems to provide this initial polarizing cue. And so this is just a list of people, current and past, that have helped with lots of efforts in the lab and collaborators and funding bodies. And I thank you for your attention and take questions if you have any.
Thank you very much, Katja. It was a very nice talk, very informative. Myself, I have a lot of questions, but I will start with the audience first. So any question from the Shireen?
Thank you for the presentation. I would like to know if this research can be used for kidney regeneration in the future.
Could this research help to kidney regeneration?
Yeah, so that's
Yeah.
sorry, that's part of what we're trying to do. So at the moment, the idea, so doing so kidney diseases and chronic kidney diseases provide a real burden on many health systems across the world. And in the adult kidney, it seems compared to other tissues that there are no resident stem cells present. So the idea how kidney repair potentially happens is that cells, tubular cells, de-differentiate to a more developmental sort of state and then grow and potentially then have to repolarize. So what we can also, we haven't done, well, we have done some preliminary studies, but what we want to do as a next step, for instance, in the organoids is to use injury models, so either hypoxia or cisplatin treatment to injure the tubules and then see what happens to the cells. Do they differentiate as the state they go to look similar to what we see during the developmental progression? And then can we help cells reintegrate better? So expression of PAX8 could, for instance, help cells to polarize better again. So that's the sort of things we would like to try out in the future. Yeah.
Absolutely. Absolutely. Another question, please. Yes.
Thank you so much for this amazing presentation. I have a question about the gene mutation of the protein HNF1B and the PAX8. How does the mutation disrupt its role as a transcription factor during the necrogenesis? And is there any mechanisms we develop through organoids that help do the same job in absence of the PAX8?
You got the question, Katja?
No, can you repeat it? It's kind of slightly distorted how it arrives at. No, if you could, no, can you repeat it in your microphone because I can hear that better.
I repeat.
Can I speak louder?
Yes, please. Yes. Okay, repeat.
Maybe I can repeat.
So
Can you hear me, Katja?
Yeah, I can hear you better, Isa. Just
great. So one question is, how are the mutations for PAX8 and for
HNF1B.
HNF1B affect basically the proteins, but
is there any mechanism we develop through organoids that do the same job in the absence of PAX8?
is there a mechanism? I don't understand that question.
Alternative mechanisms.
Independent of PAX8.
Yeah.
And are there any mechanisms that are independent of PAX8? Either in the organoids or I guess in the human renal vesicle.
In case of the mutation.
Oh, in case of the mutation. So basically redundancy, is that what you're thinking about? Is there something that could take over in case of mutations? Okay.
Yeah. So for PAX8, we think PAX2 can probably compensate to some degree because PAX8 is very similar. The binding sites across the genome are very similar. But it seems in mouse there is clear redundancy between the two. There seems to be slightly less in human, but yeah, we need to look at that. So HFN1B is a factor that is downstream of PAX8 and just important for part of the nephron formation and differentiation. So that most likely can't compensate, but PAX2 is a really good candidate for taking over in case PAX8. Although we can see in our organoids that it can't completely compensate because we see loss of the tubules, but yeah.
Yeah. Thank you, Katja. Other question? Yes, of course.
Thanks so much for this amazing presentation. I just want to know, how does aging affect the capacity of renal cells to undergo MET?
How does
aging?
Aging, okay, aging.
Yeah. So we don't know, but we suspect it will have an effect. So one issue seems to be when kidney tubules, when nephron tubes have any earlier insult during development or early life, individuals, humans, are highly predisposed to later chronic kidney disease. So that is very clear from the literature and studies, which seems to suggest that the repair of tubules is quite imperfect. And that might be because they can't undergo the first EMT and then repolarization as well as they should. So that's something to look into. How can that be boosted, for instance? What really is failing in normal repair and other ways to improve this so you avoid later chronic kidney disease?
Yeah. Question. سعيد, c'est ça? عندك الميكرو. سعيد? وجيد. Wow. Sounds the same.
Thank you for the presentation. It is possible to transfer renal organoid to animals to repair some renal disease.
Is it possible to inject transplant organoid in animal?
Yeah, so there's some labs who tried, so we're not doing that, but so as I said at the beginning, there are various limitations to our system. We don't have the collecting duct system. If you want a functioning kidney,
we lost you.
you need many more from different needs. And there are studies at the moment where people try to then, for instance, implant these into mouse to see if there's any integration into the organ. So these studies are definitely underway, but not by my lab at the moment.
Thank you.
Okay. Samer Abdullah, I have a question. Yes. Samer, Alizi.
Thank you very much for this very nice presentation. I would like to ask if you think that PAX8 implication in the process of nephrogenesis can be applied also for the MET in general, for example, in other tissues, like in the digestive tube or in the liver or somewhere else.
Yeah, so PAX8 is, for instance, also expressed in the liver. We haven't looked there at all. It could be that in some other tissues where it is expressed early on at the right time, it could have a similar effect. But I think otherwise it will just be that another related factor will switch on an MET program as the most upstream activator. But it could be, and we haven't looked into that, that PAX8 can also do that in certain other tissues where it's expressed at the right point.
Okay, thank you. Thank
Okay.
you.
We have a question from Isabel. Yeah.
Hi, Katja. Related to that, I come here so you can hear me better. How general do you think these findings are for MET events? Is it kind of serendipity? You know, PAX8 get activated, it works, it induces CDH6, and that works. Or is it something specific about this specific MET compared to others? And also how complex the network is. So, for example, could you express PAX8 independently of Wnt, and then you don't need Wnt anymore? Is that all it is? Or is there anything else? So comparative and also complexity.
Yeah. So that's really interesting. I don't know. I mean, we could try that to some extent in the organoids, see if we overexpress PAX8 early without the Wnt signal, and see what do we get transcriptionally or in terms of structure that forms. We can clearly do that. What was the beginning of the question? I forgot that bit now.
The comparative.
Oh yeah. Yes. So I don't know. I mean, MET systems in vivo, people are only now really looking in detail what controls them. So a lot was done in tissue culture, but quite often in kind of spheroids, or where there's a lot of extracellular matrix in the medium, or in the what holds the organoids or structures in 3D. And once you have matrigel anywhere in the system, you have such a strong basal signal that drives polarization, which it clearly can. And in some instances in vivo, that's what happens. But you don't know. So I think you need to really look either in an animal, in Drosophila or other models, to kind of see how it works there, or organoid systems where you not provide an overriding cue that really will just drown out anything else in terms of signal.
Okay, so then last question. Myself. It's a technical question, really outside maybe of the topic, but I saw that the cell and the organoid grow very fast. And I'm wondering, did they need vascularization in a certain moment, and when?
Yeah, so it's in the way we use our system is not too bad. So this is what was called initially a microorganoid protocol. So we only aggregate 1,000 cells. So even at the last time point we looked, day 24 usually, there doesn't seem to be any cell death due to exclusion of nutrients from the center of the organoid. But if you grow larger versions of it or other tissue versions of organoids, that can be a real issue. But there a lot of, so there's somebody in the institute who tries for various different organoid systems that different labs use to improve vascularization of these, because yes, that will help a lot.
Yeah. True.
Definitely for the functionality, in the end, we would need vascularization as well, but also for organoid survival, just at some point you need it if they become too big, definitely.
Definitely. Okay, Katja, Röper, thank you very much. It was very important.
Thank you for having me.
Very nice, very nice.
Thank you
again, and a warm wish from all the audience to, and happy birthday to your son.
Oh,
And
thank you.
hope to see you soon in another circumstance and another meeting and conference. You want to say something, Isabel?
Just to remind people that these were the exact organoids we looked at yesterday with limited fluorescent resolution, but you could see those substructures that were scaled differently, and those exactly the ones that she was showing there.
True. Because we were seeing your organoid.
Okay, great. Yeah.
Yes.
Your organoid.
Your
Okay.
Katja, thank you very much.
Okay. Thank you very much.
Excellent day.